Hello, and welcome to the Chapter 16, Part 2 lecture on DNA replication, storage, and mutations. You should use the information in this lecture to complete the Chapter 16, Part 2 guided notes, which of course you should complete before you come to class. Cells can reproduce in several different ways. In the most common form of reproduction, the cell will split itself into half and make identical copies of itself. In order to do this, it has to make copies of all of its pieces of DNA and then pass those copies on to two daughter cells so that the two daughter cells are identical to one another and they're identical to the parent. Now, in order to make that process work, the cell has to copy all of its DNA, and that's completed through a process called replication. The overall process of DNA replication is actually pretty straightforward. Let's say that we want to make a copy of this little piece of DNA here. How can we do it? Well, the first thing we have to do is open up the two strands. So the hydrogen bonds that hold the two strands together are going to be broken, and this little space in between them is called the replication fork. That's where all the action is going to happen. Next, we need to add new nucleotides to match the old nucleotides. That's going to be accomplished by several enzymes, but the primary one is called DNA polymerase. Now, there are going to be nucleotides floating around free in the cell, and what polymerase does is it gathers them up and it adds each nucleotide to its complement. So here, for instance, we have a G, a guanine. It's going to be matched up with its partner, a cytosine. Here we have a thymine. It's going to be matched up with its partner, adenine. And the same thing is going to be happening on this other strand over here. So what polymerase does is it builds two new complementary strands and matches them to the original strands. In the last step, enzymes are going to come along that kind of glue everything together. They reassemble the covalent bonds that hold together these new backbones, and they reassemble the hydrogen bonds between those strands. What you can see when we're finished is that we have two pieces of DNA that are identical to each other and are identical to the original strand that we started with. This type of replication is known as being semi-conservative. So what does that mean? Well, let's say that we wanted to copy this piece of DNA. There's the two strands. We're going to label these as O for original or old strands. In order to copy them, we need to add two new strands. So here's a new strand and here's a new strand. And these are going to be complementary to the old strands. So this guy's new and this guy's new. Because each piece of DNA that results from this replication contains both an old and a new strand, we say that the process conserved some of the old DNA but added some new DNA to it. So the process overall is semi-conservative. Remember from the first part of the chapter that DNA has directions to it. The nucleotide monomers that make it up have certain directions. Each strand is going to have a 3' end and a 5' end. And because the two strands are anti-parallel to one another, they're going to have opposite 3' and 5' ends. Now, DNA replication is carried out by a series of enzymes, and these enzymes have to grab onto the DNA and will only work in one direction. Most of the enzymes that are going to be involved in this process, including DNA polymerases, which actually do the copying, can only work in the 3' to 5' direction. That means that if we want to copy this piece of DNA, an enzyme such as DNA polymerase is going to have to grab onto this end of the DNA, and it's going to copy this strand in this direction. Meanwhile, a second enzyme is going to be required to grab the 3' end of the other strand, and it's going to be copying in the opposite direction. So it takes two sets of enzymes to do this, and they're going to run in opposite directions. They start at the 3' end of a piece of DNA and work towards the 5' end of that piece of DNA. This 3' 5' stuff can be really confusing. Because while the DNA is going to be read in the 3 to 5 prime direction, new nucleotides are going to be laid down by the enzyme in the opposite orientation, from 5 prime to 3 prime. Let me show you what I mean. Let's say we want to copy this little piece of DNA over here. To do that, an enzyme is going to have to grab onto the 3 prime end of this strand, and it's going to be working in this direction, from 3 prime to 5 prime. However, as it lays down nucleotides, because the two strands are anti-parallel or upside down to one another, the new nucleotides that are laid down are going to be in the 5 prime direction. Take a look at this pair right here. This is the 3 prime side of this nucleotide. Straight across from it, the nucleotide that's bound to it is in the 5 prime orientation. Here the enzyme is adding another nucleotide, and you can see it's in that same 5 prime orientation. So while the enzyme runs in the 3 to 5 prime direction on the old strand, it's building the new strand in the 5 to 3 prime direction. Because the two strands of DNA are anti-parallel to one another, or opposite orientations of one another, and because the enzymes that copy the DNA have to run in a single direction, it means that the two strands in a piece of DNA have to be copied in opposite directions, and that makes things a little more complex. Let's start by looking at this little area here. This area is called the replication fork. This is where little enzymes that we'll look at in a minute are opening up the piece of DNA so that copying, replication can happen. Now let's examine this strand over here, which is going to be called the leading strand. Enzymes attached to this strand will attach to the 3 prime end and will work towards the 5 prime end. The replication fork is going to continuously open in front of it. So that means that this little enzyme can just lay down new nucleotides, no problem. The fork is going to open ahead of it, so it can just continue to do so all the way until it hits the end of this piece of DNA. For this reason, we call this the leading strand, and it actually has the easiest job. I like to think of DNA replication as being kind of like helping someone move a couch into their new apartment. If you've ever had this experience, you know that one person gets to walk forwards with the couch, while the other unlucky person has to walk backwards while carrying that couch, and usually there are stairs involved. The lagging strand of DNA is the unlucky person who has to carry the couch backwards. Because its orientation is opposite to the leading strand, it means that the enzymes that replicate it have to work from the 3 to 5 prime direction on that strand. And because the replication fork is opening behind it instead of in front of it, it means that it can only lay down new nucleotides in little chunks. It might have laid these nucleotides down first, and then it had to wait for the fork to open behind it. Then it can come back and it can lay down these guys, and then it waits, and it can lay down these guys, and here it is laying down some new nucleotides again. The fork is now open behind it so it has room, and now it's going to be laying down these nucleotides. So the lagging strand works backwards, and it has to lay down the DNA in pieces, which we call Okazaki fragments. That means that the lagging strand has a more complicated job, and it tends to be a little bit slower than the leading strand. That's why it's called the lagging strand. DNA replication is carried out differently in prokaryotic versus eukaryotic cells, and of course this process is much simpler and much more straightforward inside prokaryotic cells, as are most processes. Why is it so much simpler? Well, there's two reasons. First, prokaryotic cells contain a lot less DNA than eukaryotic cells. Prokaryotic cells, on average, only have to copy about 4.7 million base pairs, or 4.7 million letters, as compared to the several billion letters you'd find inside a eukaryotic cell. The second reason that prokaryotic replication is simpler than eukaryotic is because of the organization of the DNA inside the prokaryotic cell. Inside the prokaryotic cell, the DNA is organized into a single little circle that's just called the bacterial chromosome. To copy this little chromosome, all the cell has to do is send two sets of enzymes to make two replication forks. They'll zip around the circle and copy it in opposite directions, and eventually this will separate the two circles from one another. There's an original and there's a copy. They're able to do this very, very quickly, and this accounts for the reason that many prokaryotic cells can breed a lot faster than eukaryotic cells. The average E. coli cell, for instance, can make several thousand copies of itself in just a few hours in the right conditions. DNA replication is much more complex inside eukaryotic cells than in prokaryotic cells. There's a lot more DNA inside a eukaryotic cell than in a prokaryotic cell. For instance, in human cells, there's something like 8 billion base pairs or 8 billion letters that all need to be copied. Also, eukaryotic cells organize their DNA much differently than prokaryotic cells. The DNA is laid out in these long stretches or strips called chromosomes. And inside a human cell, for instance, there are 46 of these chromosomes that need to be copied. To carry out a project on this scale, it requires a lot of enzymes and they all have to work together. And what they'll do is make multiple replication forks along a chromosome. The forks copy towards one another, and eventually this will split the chromosome in half, leaving you with two copies of that chromosome. Now that you understand the overall process of DNA replication, let's dig into the nitty-gritty. We're going to look at the enzyme machines that make eukaryotic DNA replication happen, and there are several of them. The first step in DNA replication is forming this replication fork and actually opening the DNA so the other enzymes have room to work. There are three sets of enzymes that are important to this process. First of all, we have helicase. Helicase is responsible for breaking the hydrogen bonds between the bases and opening up this fork area. So that's helicase. The name helicase tells you what it does. It unwinds the DNA helix. Now, once this fork is open, you want it to stay open. You don't want it to reseal. So that's what these little guys do. These little proteins are known as single-strand binding proteins, and I like to picture them as being like little thumbtacks that hold open the two sides of the DNA. Now, as we're pulling on these pieces of DNA down here, the area upstream has a tendency to twist, and if it twists too much, it can break, which is a problem. So to prevent that twisting, we have this third guy. He has a big long name. His name is topoisomerase, but it actually prevents this upstream area from breaking as this process continues. The next step in DNA replication actually gets the old DNA ready to receive new nucleotides. We call this process priming the DNA. I think of it as being kind of like painting a wall. If you're going to cover up an old color with a new color, it's a good idea to put down a primer coat first. This is a layer of white paint that covers up the old color and it helps the new color stick to the wall. In a similar way, we're going to prime the DNA and get it ready to receive those new nucleotides. The enzyme that primes the DNA is called primase, which is a convenient name. Here you can see it at work. It's laying down these nucleotides from the 3' to the 5' direction on the old piece of DNA. But instead of laying down DNA nucleotides, it's actually going to lay down RNA nucleotides. So it's laying down a short piece of RNA that is complementary to the old piece of DNA. Now, why is this necessary? Why can't we just put DNA directly on DNA? Well, that has to do with the shape of the next enzyme, these things called DNA polymerases that actually do the Xeroxing. Those enzymes have difficulty grabbing onto the DNA and doing their work unless there's some kind of a new nucleotide laid down already. So by laying down these RNA nucleotides, we're going to allow the next enzyme to grab on and to do the work and to put in real DNA. The process of priming and copying the DNA is a lot simpler on the leading strand than on the lagging strand. On the leading strand, really only a single primer needs to be laid down on the original 3' end of the old strand. And then the next enzyme that actually does the Xeroxing can just zip along that strand towards the fork and lay down DNA nucleotides. So it's simple, quick, it's pretty easy. However, on the lagging strand, things are more complicated. Because the primase can only work away from the fork, it means that it has to lay down primers in chunks. And it means that it leaves gaps between the primers that are going to need to be filled in. This next step in DNA replication is known as elongation of the DNA strand. This is the actual Xeroxing or copying of the strand, where DNA is going to be matched with DNA. Now, in general, the enzymes that are going to help us with this step are known as DNA polymerases. There are at least 11 of these guys that have been discovered, and there's 2 that are going to help us in this process. What's the first job that needs to be done here? Well, remember that on the lagging strand in the last step, primers were laid down in little chunks. So what we really need to do next is put in proper DNA in between these chunks to make one continuous strip of nucleotides. That's what this enzyme, known as DNA polymerase number 3, is going to do. Here you can see it in action. It's going to grab onto the 5' end of the primer from the last step, and it's going to truck along until it hits the next primer. And it's going to be filling in DNA nucleotides in those spaces. This results in little chunks of DNA existing between the primers, and we call these Okazaki fragments. DNA polymerase number 3 is also going to do our Xeroxing or copying on the leading strand. But because it's working towards the fork, all it has to do is grab onto that first original primer and then zoom towards the replication fork, laying down nucleotides behind it continuously. It doesn't have to lay them down in chunks, and it doesn't make Okazaki fragments. The next thing we need to do is take out all those primers that were laid down and replace them with proper DNA nucleotides. That's going to be the job of this enzyme, DNA polymerase number 1. Here you can see it in action. What it's doing is actually grabbing onto any little primer RNA nucleotides that it finds and replacing them with DNA nucleotides instead. So it will work from this end of the primer to there, release. It'll swing back towards the fork and find the next chunk of primer, and it will come through and replace that. Once it's done there, it'll find the next primer, and so on until it's all replaced with nice DNA. Now, there's one more enzyme that will help us finish out this process. It's called ligase. To ligate something means to stitch it together or to stick it together. So what DNA ligase does is it runs along this strand and it makes sure that all of the nucleotides have been bonded together properly. It makes sure that the shape of the DNA is uniform, that there aren't any mistakes that have been made, and it reforms all the bonds. DNA polymerase 1 also helps us finish out the leading strand. If you remember, there's a little primer that was laid down at the beginning of the strand to get the whole copying process started. And so now DNA polymerase 1 will need to come along, pull out those primers, and replace those with good DNA nucleotides. This process is actually going to be occurring in 2 replication forks within each bubble that's created in the DNA. So these replication forks will be working in opposite directions, and eventually they will reach the end of the chromosome, at which point the chromosome will be split in half. So you'll have 2 complete copies of the chromosome being made. This model of DNA replication shows you the parts played by all the different enzymes involved in the process. But to be honest, this model's a little bit deceptive. And the reason is that it shows the enzymes as being totally separate from one another, working one after the other to make this replication happen. In reality, all of these enzymes are complexed together into one giant machine, and they kind of work together all at once to copy the DNA. This machine is really cool looking, and I want you to get a look at it. So on the next slide, I've posted a video that's going to show you what this machine might look like if we could see it happening inside the cell.
Using computer animation based on the latest research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
Here's a startling fact. Your DNA actually gets a little bit shorter every single time it's copied. And in some types of cells, this copying can happen hundreds or even thousands of times a day. Why is that happening? That sounds scary. Well, it has to do with the way that DNA polymerase grabs onto the DNA during replication, and it's just not able to grab this last little segment on the end of the chromosomes. So that RNA primer that's been laid down on that last little segment gets snipped off, and the unmatched little segment of DNA that was attached to it gets snipped off too. That happens at both ends of all of your chromosomes, all 46 chromosomes inside a cell. So the ends of these get snipped off and the chromosome gets shorter. If the constant shortening of your DNA scares you, don't worry, you have a safety net built in. On the ends of all of your chromosomes, you have these sections called telomeres. Telomeres are long sections of DNA, so they are made of A's, T's, C's, and G's, but they don't code for anything important. They're essentially just junk DNA. So they're not genes, they're just some letters that have been thrown on there. Because they don't code for anything, these telomeres can safely be snipped off a little bit at a time every single time your cells replicate and reproduce without chewing up the good DNA in the middle. Now, that being said, it is true that each different type of cell can only replicate itself so many times before you do start to chew up good genes. Because cells are limited in the number of times they can reproduce by the length of their telomeres, telomeres have been implicated in the aging process. Now, scientists don't really know what the connection really is between telomeres and how long we live, but there's a lot of really neat science coming out of that exploration. For instance, what if we could take our telomeres and rebuild them? If we could add to the telomeres, could we sort of reverse the aging process or lengthen our lifespans? It's an interesting question. There is an enzyme that is known to do this. It's called telomerase, and it's only active normally in embryos. Imagine what's happening in an embryo as it grows a body. It has to grow all different types of cells and organize them into organs and organ systems. So there's a whole heck of a lot of replication and reproduction going on. To compensate for the shortening of the telomeres, telomerase is active in the embryo and adds length to those telomeres so that the cells can continue to replicate at such a high rate. What if we could turn this on in a grown adult? Again, could we add years to our lives? Could we reverse the aging process? Scientists don't really know, but they're definitely looking into the question. Every time that a cell copies its DNA, It has to add the complements to millions or even billions of letters, and it really has to do it perfectly or it could develop problems. There are enzymes that are responsible for double checking this copying process. To some degree, the DNA polymerases do this, and there are also enzymes known as nucleases that go back over the DNA and read it and make sure it's been copied properly. However, mistakes do happen in spite of this. We call mistakes in the DNA mutations. Mutations are random changes to the DNA sequence, but they are oftentimes permanent changes to that sequence. Here, for instance, there was a guanine in the original sequence and it's been changed to an adenine. And here's another mutation. There was a thymine in this sequence and now that's been changed to a guanine as well. Mutations can cause problems for cells because they change the information that's actually stored in the DNA. So they can make changes to the proteins and RNAs that are built based on that information. By far the most common cause of a mutation are simply errors in the copying process. The machinery of the cell that carries out DNA replication makes a mistake and sticks the wrong letter in at the wrong position. Remember that anytime a cell goes through DNA replication, it has to carefully, even perfectly copy millions or even billions of letters at a time. Now, even given that, cells don't make mistakes very often. But when you think about the number of times they have to do this in an average day, you can get quite a few mutations. Think about this. Bone marrow cells make red blood cells by dividing. They can divide as many as 150 billion times a day to make a new ounce of blood. So within those 150 billion times, and think about the 8 billion letters they're copying each time, there's a lot of potential for making mistakes. Another potential cause for mutations is environmental damage. Typically, this is exposure of cells to things like UV light, as in this poor gentleman, or really nasty chemicals, things we call mutagenic chemicals. Exposure to these things can directly disrupt the DNA, or it can affect the machinery that works with the DNA so that they make more mistakes. This is the reason that exposure to UV light, that tanning, can cause cancers. That UV light can affect the DNA of a skin cell. That skin cell can start to grow out of control, and it can cause a tumor, a cancer. There are many different types of mutations, and they can affect DNA in different ways. There are several different ways to classify mutations as well. One way to classify mutations is based on the amount of DNA that they affect. If a single set of letters is affected, we call that a point mutation. Now, even though point mutations only affect one pair of nucleotides, they can be really powerful, and we'll see an example of that later on. Another way to classify mutations is based on how they affect the DNA. For instance, in this mutation, this pair of guanine and cytosine has been swapped out for an adenine and its complement thymine. Because the letters have changed, we call that a substitution. Mutations known as insertions and deletions also affect the nucleotide sequence. In an insertion, a nucleotide pair or pairs is inserted into the sequence, so letters are added to the sequence. Here, for instance, we have the sequence CTT, and you can see that an insertion mutation has occurred here because now the sequence is CATT. A deletion is the opposite of an insertion, so it's the removal of a pair or pairs of nucleotides. Inversions and translocations tend to be larger scale events, so they tend to happen with more than just a single pair of nucleotides, although they can occur with a single pair as well. In an inversion, a section of DNA is cut out, flipped over, and glued back into the sequence. And because the two sides of DNA are not identical to each other, this is going to change the sequence of both strands. In a translocation, a section of DNA will be cut out carried somewhere else in the cell, and glued into another sequence. And that, of course, can change the sequence as well. Another way to classify mutations is based on the type of cells that they affect. Somatic mutations occur in what we call somatic cells. These are body cells that are not reproductive. So somatic mutations can occur anywhere in the body other than the testes or the ovaries. So let's say that a beam of UV light comes down from the sun and it strikes this skin cell right here on this guy's side and causes a mutation that causes a cancer or a growth. Now that's bad news for him. He's going to have to deal with that cancerous growth. But the good news about somatic mutations is that they aren't passed on to the next generation. Because it happened in a little skin cell right there on his side, and that skin cell is not going to be passed on to his kids, he's not going to pass that cancerous gene on to his kids. Germline mutations occur in cells we call gametes. Gametes are sex cells, so gametes include the egg cell and or sperm cells. Now, germline mutations can be a little bit more insidious because remember that a fertilized egg is going to divide and divide and divide, and it's going to build the body of the offspring. So a mutation that occurs in an egg cell, for instance, is going to be passed on to the next generation. And that mutation will occur in every single cell of that offspring's body. So germline mutations can be passed down from mother to child or from father to child, and can cause problems in the offspring. Incidentally, if you've ever gone to the dentist and had to get x-rays, you might notice that they make you wear this giant lead apron. And you might have also noticed that that lead apron does go down over the lap area. The reason they're making you wear that apron is to prevent this type of mutation. They're going to zap your body with x-ray radiation. That can potentially cause mutations, and they want to make sure that your internal organs, including your reproductive organs, are protected from this. So how often do these crazy changes occur? Well, scientists estimate that about 1 in 1,000 nucleotides are incorrectly copied every single time the cell goes through DNA replication. And when you consider the number of divisions that our cells go through during a day, and when you consider the lifespan of a human being, these somatic mutations can really build up over time. Scientists also estimate that every single human being is born on average with 60 germline mutations. So those are mutations that occurred in the sperm cells and the egg cells that conceived each of us. And so in a sense, we are all mutants. Not in the cool X-Men kind of way, but in more of the realistic scientific kind of way. Incidentally, I should mention that not all mutations are bad. We often think of mutations as being associated with diseases like cancer, but most mutations are neutral and don't affect the body at all, or mutations can even be beneficial. This is what drives evolution. For instance, a person might carry a mutation that changes an enzyme so that they can eat a new food source and have access to that. So not all mutations are bad. Finally, we need to discuss how DNA is stored inside your cells. Just to remind you, your DNA is split into these long sections called chromosomes. So a chromosome is just a long piece of DNA. If I were to look inside a nucleus of one of your body cells, I would find 46 of these chromosomes. 46 is the magic number for human beings. Different organisms have different numbers of chromosomes. Now on those chromosomes, I'm going to find the individual recipes for making specific proteins or RNAs. We call those specific recipes genes. So a chromosome really consists of a bunch of genes that are strung together with some other little segments of DNA in between, which we'll talk about in a later chapter. Consider this. If I took the DNA from a nucleus of one of your cells, if I took all 46 chromosomes and stretched them out end to end, I would have a molecule that was nearly 6 feet long. And yet all 6 feet of this DNA can be crammed inside a nucleus that is far too small to see with the naked eye. How is that even possible? Well, it's all in how the DNA is stored. I want you to think of DNA as being kind of like yarn. If you've ever worked with yarn for a crafting project or a knitting project, you know it can really kind of be a mess. You don't want it to just be stored randomly or willy-nilly. You want your yarn to stay organized so it's easy to work with. So with yarn, you take the yarn and you ball it up. You either ball it into a skein or a nice tight little ball. DNA does something kind of similar. Here is linear DNA. It's like a long piece of yarn. That long piece of yarn will be carefully wrapped around these proteins called histones. So it gets wrapped around and around and around these guys, and that helps ball them up. Collectively, a histone and the DNA that's wrapped around it is called a unit called a nucleosome. That fiber will then be twisted up on itself a little bit more to form something called chromatin. Chromatin is the working form of DNA. Most of the time during its life cycle, DNA is going to be in the chromatin form. It's loose enough for enzymes to get in and work with it, but it's not so loose that it's disorganized or going to get damaged.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Hopefully this chapter has provided you with a little bit more information about DNA, about how it's put together and how the cell uses it. We're going to use this information quite a bit more in future chapters, so keep it in mind. And remember, never ask a science nerd a question that you don't want a really thorough answer to. Bring your questions to class and we'll work on them. See you next time.